Hello, everyone, and welcome to phyloseminar.org. This is the second talk in a series of 3 talks on the intersection of phylogenetics and cancer research. Please use the YouTube live comment box to ask questions. Today's speaker has been Raphael. Dr. Raphael is professor of computer science at Princeton University. His research is focused on the design and application of computational methods to analyze large-scale biological data. His group uses diverse computational approaches, including combinatorial optimization, graph algorithms, machine learning, and statistical methods. Recent areas of emphasis include cancer evolution, network pathway analysis of genetic variants, and structural variation in human and cancer genomes. His algorithms have been used in multiple projects from the Cancer Genome Atlas to the International Cancer Genome Consortium. Welcome, Ben, and thank you for participating.
All right, well, thank you very much for the invitation and the opportunity to present. It's great to be part of this series in cancer evolution. And today I'm going to tell you about some of our work on cancer evolution and looking at how tumors evolve over space and time. So by way of introduction, cancer is an evolutionary process. It's a process by which somatic mutations accumulate in the genome of some organ. And these somatic mutations are generally split into 2 classes, those that are just sort of passenger random mutations, which don't have any effect on the cell, and driver mutations, which might cause the cell having them to evade the normal controls on cellular growth and grow quickly and start to form a tumor. And so you tend to have these clonal expansions in a tumor. And so when we measure a tumor at the present day, we'd see that it's a very heterogeneous mass of various types of cells, both normal cells from the tissue of origin and cancerous cells that have somatic mutations that they've inherited during this evolutionary process. And so this sometimes sort of visualized in this picture on the right, where you can sort of see these clonal expansions, and this is sort of illustrating this sort of population of each clone. But like any evolutionary process, you can also describe this via a phylogenetic tree. So here the vertices in the tree are individual cells, or sometimes groups of cells called clones that have a shared set of mutations. And on the edges, we can indicate the somatic mutations that have accumulated and distinguished the parental cell from the child cell. So evolution, tumor evolution, is really key to understanding and treating cancer by building trees that represent the evolutionary process over different individuals. We might see common patterns. We might see mutations that are on a trunk of the tree, ones that are early in the process, mutations that tend to be closer to the leaves of the tree, so-called subclonal mutations, which tell us something about the dynamics of this evolutionary process. We might also look at how the tumor has evolved and how cells have left the primary tumor, metastasized to distant organs, and seeded new tumors at these distant organs. And understanding this metastatic process is also key, because metastasis is really the major ways that cancer leads to death. Even more precisely, looking at an individual tumor, we might be able to see, by understanding its phylogenetic tree, where mutations are that, again, are present in all the cells. These might be ones that we can target for particular therapies. We might be able to actually use sort of different constraints that are given to us by the evolutionary tree, knowing that we have to maybe target different subpopulations distinguished by different branches on the tree, and even thinking about how we might adapt therapy to the subpopulations of cells and how they evolve over time. So all this really begins with building the tumor phylogeny. And this is a classic phylogenetic problem, where you would take a tumor sample at the present time, you would sequence it. Since we want to measure these somatic mutations that are in the genome, we perform DNA sequencing. We would then get some data giving us, for the individual cells we measured, the mutations and whether or not those mutations were measured or not in that cell. In this case, I'm sort of indicating a mutation that might be a single nucleotide change in the tumor genome with respect to the inherited parental genome of that individual. And then from this mutation matrix, one could apply one of the many phylogeny construction algorithms to build this phylogenetic tree. So what then are the challenges in performing this process with tumors? Well, the first major challenge in studying cancer evolution is that there's really many technical limitations in the technology for measuring somatic mutations. And this is because when we're looking at tumor evolution, we're looking at evolution at the level of individual cells. And so what we need to measure in order to build this matrix is the mutations in single cells. These are our taxa, our leaves of the phylogenetic tree. And single cell DNA sequencing just remains very difficult technically. And so often what this leads to is that we need to construct a phylogeny from a mutation matrix like this that has large uncertainty in the mutations due to the limitations of our measurement technology. And so we have to think about a lot the technology we're using and what it tells us about the data that we have. And in terms of DNA sequencing and RNA sequencing, there have been two technological revolutions in the technologies for measuring tumors. Until about 10 years ago, most of the tumor sequencing that was done was of a single bulk tissue sample. So you would take out a part of a tumor, you would then sequence this, and you would get a single aggregate measurement of all the cells in that sample. Around 10 or so years ago, technologies to barcode individual cells and then measure the mutations in those individual cells in a massively parallel way started to come online. And so these single cell technologies allow us to sort of get maybe hundreds or thousands of cells and the mutations in those cells measured, but with some limitations, which I'll discuss briefly. And then even more recently, in the past few years or so, there have been technologies to actually adapt some of these type of barcoding technologies to now barcode not individual cells, but individual locations within a two-dimensional tumor slice. And so with these spatial sequencing technologies, we can measure not just in the mutations, but also where those mutations are within the physical space of the tumor. So the second major challenge in tumor evolution beyond these technologies is that the somatic mutations that occur in the cancer process occur across all genomic scales. So at the simplest type of mutation, there's the single nucleotide change that I sort of mentioned, but the changes go up the scale from small insertions and deletions, larger gain or loss of segments of the genome, rearrangements of the genome, whole gain and loss of entire chromosomes, and even the whole genome duplicating, creating its extra copy of every single chromosome in the genome. So now when we measure or sequence a cancer genome, we would measure mutations across all of these scales. And we have to think a lot about what are the markers, which mutations do we want to use of our markers of lineage or evolution when we build our tree. And depending on which ones we use, we might have to use some very specialized evolutionary models or evolutionary distances, which I'll discuss. So I wanted to talk about these challenges in the context of two more specific problems. First is how we construct phylogenies from longitudinal cancer sequencing, where we might have sequenced a tumor from one individual at multiple time points. And second, how we might study the evolution of a tumor over space, and actually do this over two scales. One, the scale of an individual, where the primary tumor leaves the bloodstream and seeds metastases at different sites. This is a form of spatial evolution. And then also at the micro scale, where we're looking within a single tumor sample and looking at how that tumor has grown and mutated at the scale of micrometers. So to begin with the longitudinal part, we think again about the different technologies we have available to us. And most of the data that was generated, in fact, most of the tumor sequencing data that's probably still generated is done from bulk tissue samples. Right? So this is, again, we're going to measure this aggregate of all the cells that we put into the DNA sequencer. So I'm going to start by describing some of the challenges with that type of data and how it also bleeds into related challenges in single cell sequencing data. So if we take a sample from a tumor, this sample would be thousands to perhaps millions of cells, and we sequence the DNA in those cells, Then what we'll have are, on a short read sequencer, sort of a Illumina sequencer, billions of short DNA sequencing reads from all the cells that made up our sample. And we won't know which read came from which individual cell. So now we don't have any notion of which mutations are present in which cell. We only have a notion of which mutations did we measure in each sample. But it turns out we have a bit more information, because if we look at all the reads that align to the reference genome when we're calling a mutation, so in this example, I show sort of 5 reads that are aligned to the reference genome at this location. In the reference genome, there's a G, and 2 of these reads have a T, indicating there's perhaps been a mutation to a T at this location in the genome. And what we can actually then try to derive is, based on the number of reads that contain this mutation, this gives us an indicator of the fraction of cells that contain this mutation. So we can actually get slightly more information. In each sample, we have not only the presence or absence of a mutation, but we have some measurement of the fraction of cells within that sample that have that measurement. So now, instead of a mutation matrix, maybe 0 or 1 valued presence or absence, we instead have this frequency matrix with these values between 0 and 1. And so this creates a very interesting phylogenetic problem I sort of refer to as phylogenetic deconvolution, where if you have 1 or more samples from a bulk measurement, then for each of the mutations, you'll measure in each sample some notion of the fraction of cells that have that mutation. And now, from this frequency data, can you recover the evolutionary history of the tumor from this mixture? So as sort of an analogy, we think that in traditional phylogeny, we measure individual species at the leaves of our tree. We measure the mutations in these species, here just indicated again with 0s or 1s for presence or absence of the mutation. So we have the leaves measured of an unknown tree. In this problem, we instead have sort of mixed all our species together. So maybe we have the frequency of each mutation present on each of the continents. And now we would have, could we build our tree from mixtures of unknown leaves of the unknown tree in unknown proportions? So actually it's quite a challenging but interesting problem. I'll talk about that in a second, but I wanted to first show the related problem that happens with single-cell DNA sequencing data. So with single-cell DNA sequencing, we extract individual cells. We might barcode these cells, but because we're sequencing the single DNA molecule that makes up the genome of that cell, it's a very, very difficult signal. So usually we have to provide some amount of genome amplification to amplify that single DNA molecule from that single cell into a signal that we can sequence on sequencer. And then we do that, and we ideally would obtain a measurement of each cell and each mutation, whether or not that mutation was measured in that cell. But because of the various errors that can happen in both of these processes, the amplification process, the extraction process, the sequencing process itself, we don't get this perfect 0-1 matrix. Instead, we get a matrix that has high rates of missing data and some false positives and some false negatives. So in many cells, we'll fail to measure the mutation, or we might measure the mutation incorrectly. And these errors are, again, due to the various techniques we have to apply. When we do this genome amplification, we have various biases in how uniformly we can amplify. And so that will lead to what's sometimes called allelic dropout, where we don't measure one of the 2 parental chromosomes. We might fail to measure both the parental chromosomes. We might, when amplifying, introduce errors, or when we sequence, introduce errors. And so all of these lead to errors in this matrix. So this creates another variation of the classic phylogeny problem, which I refer to as the phylogenetic imputation problem, where now in this problem, we've measured the individual leaves of our tree, the individual cells, but we've measured them with some amount of noise. And so can we build a tree from this data with noise? So we've got these traditional phylogeny where we measure the leaves, but we don't know the tree. And then we've got these two variations where we measure the trees with noise in the single cell sequencing case, or where we don't measure the leaves, we instead measure mixtures of the leaves in the bulk case. So how can we try to solve these two variations of the traditional phylogeny problem? And so the approach that's been taken in the cancer community is to impose a evolutionary model on the mutations and then try to actually build the tree under this model while correcting or deconvolving the data. And to begin, in order to do this, we'll use, and what has been done in the field is to use the simplest evolutionary model, which is so-called perfect phylogeny model. Right. So in the perfect phylogeny model, the simplest evolutionary model there is, we imagine that our phylogenetic characters have only one of two states. So each character or mutation is either mutated or not in a cell. We'll assume that we have the ancestor. In this case, the ancestor would have none of the somatic mutation. So it would be, in our case, the all zero cell. And then importantly, we're going to assume that a character changes state at most once in the whole evolutionary history. So what that means is that our phylogenetic tree has an extremely simple structure. So each leaf is labeled by a species or one of the rows of this matrix. The root, again, is the all zero. And then every character, because it can change state at most once on the tree, actually uniquely labels one of the edges of the tree. So in this example, I've labeled the label the edges with a character that has changed from 0 to 1 along that edge. So what that means again then is that for each leaf, because on the unique path to the root, we can trace out the characters that have changed state. So leaf A here, as I trace out the path to the root, has changed state for character 2 and character 1, and we see then that it has the ones here at character 1 and character 2 and only those two, etc. So this perfect phylogeny, this no homoplasia assumption, is also in population genetics sometimes called the infinite sites assumption. So now, under this assumption, there's a very simple and straightforward characterization of the tree and a very simple and straightforward algorithm to build the tree from the data. Turns out you can look at the 0-1 matrix and you just have to check, is there a tree that will have the leaves labeled by the rows of this matrix? There is, if and only if no pair of columns contain all three pairs of 0, 1, 1, 0, or 1, 1. This is the so-called three gametes condition. So I can look, for example, here at columns 3 and 4, characters 3 and 4. I see the 0, 0, the 1, 0, and the 0, 1, but I don't see the 1, 1. So columns 3 and 4 meet this criteria. And you can check that all pairs of columns meet this criteria. And with such a matrix, we can build the tree in time that's proportional just to the size of the matrix. Okay, so this is the simplest phylogeny problem. Now, interestingly though, when we take this simplest model and now we mix it, so we look at the corresponding problem where we've measured not this mutation matrix, but instead measured these frequencies, as we would have in bulk sequencing, then our problem becomes to deconvolve this matrix, i.e. find the tree and the leaves of the tree, which when mixed together gives us this frequency matrix. And about a decade ago now, we posed this as a matrix factorization problem, and we showed that this was a NP-hard problem. We derived an algorithm to solve it using an integer linear program. Prior to that, there were many, many tools that were solving this problem using a variety of heuristics. So trying to turn these frequencies into a phylogenetic tree. All right, so in bulk, we have this problem of matrix factorization for the perfect phylogeny matrix. In single cell, where we have errors and missing data, we can now pose that as the problem of matrix correction. So we try to take our data, our mutation matrix with error, we try to find the perfect phylogeny matrix that's closest to our measured matrix. So this is a problem now that we think about how would we do this? How difficult is this? Again, even under the simplest model, the perfect phylogeny model, we find that this is an NP-hard problem if we asked for the minimum number of changes or edits we need to make to the matrix. So we need to fill in each of the missing data, each of the question marks with either a 0 or 1, and we might have to change zeros and ones and correct them when they're errors. And if we try to do that with the minimum number of edits, this is also an NP-hard problem. So in this case of the simplest evolutionary model, the model where we can build the tree efficiently in polynomial time, both the deconvolution problem and the imputation problem become NP-hard, difficult problems. In practice, what researchers have done, and this was sort of pioneered by Florian Markowitz and Nico Bärenwinkel, who actually gave the seminar in this series last month. You use a probabilistic model. So you try to find the tree that maximizes the probability of the observed data, where the observed data here is either this matrix with errors, or in some cases, you look at the actual read count data, which I'll talk about in a few minutes. And you could try to find this under a maximum likelihood framework, or more interestingly, by actually trying to marginalize over the cell assignments, because there are typically many more cells than there are mutations that we can measure with present technologies. So just as an example of that latter approach, here's a method SciFi from Nico Bärenwinkel's group. This was applied to data where 255 individual cells from an acute lymphoblastic leukemia, a blood tumor, were measured. In this picture on the left, what you can see is the mutation matrix with the individual probabilities of a mutation being present at that location. So you can see this matrix looks very noisy, but now when you try to impose this perfect phylogeny correction, you get a matrix with a corresponding tree that has this nice sort of block form. So that's sort of the lay of the land for how people have looked at building trees from bulk or single cell sequencing data. So now what about longitudinal samples? So one of the interesting, important things in cancer is to actually monitor how the tumor is changing over time in the individual, how it might be changing in response to various treatments. And so in some tumors, particularly tumors in the blood, but increasingly solid tumors, if we sequence circulating tumor DNA in the blood, we can measure the cancer in the individual at multiple times. So this was work that was pioneered over a decade ago with blood tumors. Here I'm showing you two examples, one from acute myeloid leukemia, where the tumor was sequenced at 3 different time points. The first was pre-chemotherapy, the second post-chemotherapy, and then a later time point. And what you can see from this plot, again, each of these, the height of each of these colored things here indicates roughly the frequency of the individual clone that is found. Clone, again, distinguished by different somatic mutations. So you can see a very diverse tumor pre-chemotherapy with multiple populations of cells following chemotherapy, a tumor that seems to only have one predominant clone. And then in the relapse, the tumor, again, grows out with new mutations, although seemingly one clone seeming to dominate. More complicated story in this lymphoblastic leukemia. Now, in these early studies, these sequencing was done from just a blood sample. So this was bulk tumor sequencing, mixing together all the mutations. And so when we, years ago, were thinking about these problems with bulk data, we started applying some of the state-of-the-art tools for building phylogenies from bulk tumor sequencing now to these multiple time points. If we apply these tools to, in this case, all 5 time points simultaneously, we get some curious findings. Now, first, this problem of building out the phylogeny that is consistent with this mixture is a vastly underdetermined problem. So there are often many possible solutions, and each of these algorithms has slightly different ways of sort of scoring or prioritizing these solutions. And so in one algorithm, this was ours, Ancestry, if we look at what the proportions of each of the individual clones were inferred at each of the time points, we see that we get this very sort of boring solution in terms of the evolution, where all of the interesting evolution has happened at the first time point, all clones are present at the first time point, and then they just vary in frequency as we go along. In another approach, phylosub by Quade Morris's group, this outputs a different reconstruction, a different tree from these 5 time points. And here there's some more interesting dynamics where at the first time point, only 2 of the clones are present, and then things change over time. But I wanted to highlight this clone 1, which you can see is at 0% frequency at the first time point, grows out to 0.7% at the later time point, disappears again, and then comes back with this really rare clone at 1% at the 5th time point. So this sort of disappearance and reappearance of a clone was kind of a mystery to us. And there are 2 ways one could think of this happening, actually 3 ways. One might be that this is actually genuinely the biology. The clone disappeared, and then all those mutations came back again later in exactly the same sequence. Seems like an unlikely scenario. Another possibility is we just failed to measure the clone at these intermediate time points. And that is also possible. We're sequencing this blood sample, and certainly we don't expect to measure every single mutation, even those at really low frequency. But the third possibility is sort of suggested by the fact that when we ran a different algorithm, we get a different answer. And that's that there's just some uncertainty in these reconstructions. So what we thought about at the time was, is there a way that we can derive phylogenetic trees that are longitudinally consistent? Meaning we won't have these strange cases where clones disappear and reappear. And so several years ago, we developed a approach called Calder, sort of named after the artist Calder, because the trees sort of reminded my former students at the time of these beautiful mobile sculptures from Calder. And what this does is it solves this deconvolution problem that I sort of told you about, this matrix factorization problem. But it imposes some additional constraints on this matrix U, which describes which clones are present at which time, this sort of mixture matrix. And these conditions are applied in a way that we skew towards solutions where we don't have clones disappearing and reappearing. And this is sort of described in the paper. I'm not going to go into details of that here, but I just wanted to show you that then if we apply it on the same data, we get sort of yet another solution. Here's a solution where not all clones are present the first time. There's only sort of 2 clones present at the first time point. And then there's sort of further evolution with different proportions of different clones, but there's no strange disappearance and reappearance of a clone.
Ben, do you mind clarifying what is the longitudinal consistency that you're actually applying here?
Yes, I'm gonna do it on the next 2 slides for the single cell if I can, because this one gets more complicated. Yeah, no, this is the perfect question. So this was old work, and I figured we were kind of done with this type of thing, but then we started looking a little more detail about some of the newer data and the single cell sequencing data. So this is data now 5 years old, although there's more of this type of data that uses a technology that sequences DNA from individual cells, although it does it in a very targeted way. So you have to pick ahead of time roughly 400 genes that you want to sequence. And so if you, as we do now, know a lot about the driver mutations that are present in different tumors, we can pick the genes to sequence for a particular tumor type and then study the evolution of those tumors at the single cell level. And so in this paper, they built trees using a tool called CITE from, again, Nico Baron-Winkel's group. And I'm going to sort of explain this tree to you and then tell you about this longitudinal constraint. So because there are many cells that were sequenced here, thousands of cells, the trees are actually sort of shown with mutations as the nodes of the tree. This is sometimes called a clone tree. And then what's shown here, so we see this tree on the nodes, and then what's shown at each node is whether or not the corresponding set of mutations or clones on the path to the root were present at each of the 5 time points when it was sequenced. Okay, so if we look at the first time point, we see that only a few of these clones were present. These 3 clones right here are absent. Go to the second time point, they're still absent. The proportions of some of these clones have changed, et cetera, et cetera. But now, if we look in this red box here, we see again this issue where this clone that contains RUNX1 and all the mutations in the parent is present at low frequency here, still at low frequency, disappears, not present at the third time point, and then comes back again for the later 2 time points. So again, we have this disappearance and reappearance of clones. Now, if we think about what had happened in this analysis, this was again trying to impute this missing and erroneous data from the single cell in a way that gave this perfect phylogeny tree. There's again not a unique way to do this. And so we could ask, are these disappearing and reappearing clones really that well supported by the data? And so we needed a way to sort of write down what a longitudinally observed tree would look like. And this is what we did in recent work. This is a method called phylocron, which we posted as a preprint a few months ago. So let me describe this for you and describe this longitudinal assumption. All right, so the data we're given is going to be in the form of the actual read counts, which go into making deciding on whether or not there's a mutation. So this is actually sort of the raw data. Okay, so for each cell at each time point, and we've indicated the time points with this time point vector, for each of the possible mutations that were measured in the data, we count up how many reads are there in that cell that indicated there was that contained the mutation, and how many total reads were at that location. Okay, so this blue mutation in this first cell, there were 20 reads that covered the location of the genome where the blue mutation would be, but none of them had the blue mutation, right? So it seems like maybe the blue mutation is not present in that location. All right, so this is the total read counts, the variant read counts. And so then what we'd like to do is find a most likely longitudinal perfect phylogeny tree or perfect phylogeny matrix, because there's a one-to-one correspondence between the matrix and the tree. This matrix, again, is it just indicating presence or absence of the mutation in the corresponding cell? So we want to find the most likely one. And then we'd like to do a test to ask, what's the most likely longitudinal perfect phylogeny versus the most likely perfect phylogeny, to sort of see what is the support for this longitudinal assumption. All right, so this requires that we define this longitudinal perfect phylogeny. So this was the question that was being asked. All right, so here is the definition. So begin with definition of a longitudinal tree. So a longitudinal tree is going to be a rooted tree, in our case, where the leaves and some, but not necessarily all, of the internal vertices are labeled by time points. Okay, so these are our observed times. We just think of these as integers from 1 to k. So all of the leaves are measured, observed at the third time point, and some of the internal vertices are observed at earlier time points. And the condition that we impose is that every path from the root to the leaf has time point labels that are consecutive in an increasing order. So you can see in this version of the tree, every path from root to leaf has labels 1, 2, 3. Now note that, not every cell made it to the third time point, and there are certainly cells in between that we didn't observe, but the time points are ordered on every path. So what this requires is sort of an assumption about how we've sampled this population as we grow. And if we think about single cell sequencing, it's a destructive technology. So if we sequence a cell, we don't get that cell is gone from the population and will not be sequenced again. And so what we're sort of assuming here is that every cell that's sequenced at a later time point had an ancestor that was sequenced at the previous time point. Okay, so there's an assumption about the sampling. This doesn't have to hold. Certainly, data where we might not have sequenced every ancestor, but with this assumption, we can describe this tree, and then we can describe what then a longitudinal perfect phylogeny is. It's perfect phylogeny. So that's, again, the condition that I showed where, every mutation, it happens only once. There's no homoplasy. There's this 0-1 matrix. But then in addition, we have this condition on the time points of the corresponding tree. Okay, so that's the setup. That's the definition. And now we can ask, could we now find a longitudinal perfect phylogeny given this data? And how hard is it to do it? What's the algorithm to do that? And that's what's described in the phylocron paper here. So again, we're going to do this with this read count data, and we're going to find then the most likely perfect phylogeny matrix that has this longitudinal constraint on its time points that maximize the probability of the data. Turns out that's an NP hard problem. Not surprising, since all the other imputation problems there were NP hard. And so what we did in the preprint is we derived a way to do this, exactly. It's a mixed integer linear program, and it's done for a fixed clone tree. So the clone tree is sort of the skeleton where we have the mutations already sort of in order, and what we need to do is just attach the cells to the nodes of the tree in this way. So there's ways to sort of get these skeletons, and so then we can find the most likely longitudinal phylogeny, and then we can ask, what is the support for the longitudinal phylogeny or just the generic perfect phylogeny? We can compute the most likely perfect phylogeny using any of these existing algorithms like SITE that I showed you, SciPhi. There are several. We now have a way to compute the most likely longitudinal perfect phylogeny. These are two nested hypotheses. Every longitudinal perfect phylogeny is a perfect phylogeny. So we can look at the likelihood ratio to sort of see the evidence of one in favor of one versus the other.
Sorry, okay, you did a great job explaining the longitudinal phylogeny. I don't know if I understand about the probability of the longitudinal phylogeny.
Yeah, so the probability of longitudinal phylogeny, so it's the probability of the matrix. So this is a generative model. So given the matrix is the presence or absence of each mutation in each cell. Okay, so given this matrix and given the total read counts, it is just a generative model for what the variant read counts will be. So this is like the error model of the observed data.
I see. So should I be thinking of this as like a collection of Poissons or something? Or?
Yeah, exactly. So Poisson is a good model. Poisson, negative binomial.
Yeah, all those good things.
All of those. Any choose your favorite model for generating, variant reads from these mutations. Beta binomial has been used as well.
And okay, we do this hypothesis test. I'm just trying to understand. So what is this actually testing? It's testing like, is there a longitudinal signal in the data?
Yeah. So it's testing like, is there longitudinal signal in the data? It's really, you think of it as testing like, can you reject the longitudinal hypothesis? So longitudinal is like the null hypothesis in this setting because of this nesting. Perfect phylogeny is sort of the alternative. Longitudinal subset of that. So we're asking if you can reject the fact that the data was from this longitudinal process.
But what would that mean? Like, what does that mean? The data, we know the data is longitudinal.
Yeah. So the data is longitudinal. I described this as longitudinal sampling because in order to make this definition, we had to assume this thing about how the sampling process was done. So the data is longitudinal, but it could be that we, failed to measure a important cell at a particular time point, and so it was just a question of undersampling, and that's why we got this clone disappearing and reappearing. We just didn't measure it, and so what we're sort of doing here in the definition, we've said, okay, in essence, this is like we completely sampled everything, and then we ask, is the data actually consistent with this complete sampling? So we're sort of ruling out these, disappearance due to undersampling.
Okay, which was like, that's like going way back to the beginning of like when you were doing these things and then cells were appearing and disappearing and you're like, that looks funny. And so this is exactly a formalization of that. Okay, exactly. Thanks for bringing it back to that. Okay, yeah. I got it. Thanks.
Good. No, thanks for the questions. It's always harder online because, you don't get to see the faces of the audience and the confusion. But yeah, so that's what's happening here. We're just asking like, does the data support or, can we reject this complete sampling assumption? Can we reject this, complete longitudinal sampling assumption? All right, so now when we apply to the data set that I showed, here we sort of reanalyzed it with a more recent algorithm that does this single cell imputation. We again see this issue. Here's the different clones. Here's these sort of attachments. So again, at each time point is measured how many cells are present for that particular clone. And what we can see again, this red one here is present at the first 2 time points, disappears and reappears at the 4th time point. Here's other disappearing clones that occur. And we now run under the longitudinal assumption. And of course, then we're going to force ourselves to get solutions where there's no such violations. Now, if you can read, depending on the size of your screen, there's a small number of cells here, 4 cells here, 3 cells, 4 cells. So it's a really small number of cells that are present at these time points. And so we can do 2 things. One, we can ask like, well, did we reject the longitudinal assumption on this data? And when we do our log likelihood ratio test, we fail to reject the null hypothesis. So we would say there's not really enough support in the data to say that these cells were not present. Okay? So we would say that they are. There isn't this disappearing and reappearing clone. And then we can actually go look at the underlying data for these small numbers of cells. And that's what's done here. So for each of the cells, here is a row. Here's the corresponding mutations. And I could ask, how many total reads were at the locus for that mutation, and how many reads are there that support the mutation? So you can see this first one, for example, Compass said this mutation wasn't present, but actually there's 1 variant read out of 42 total reads. Everything in green has some variant read, but you can see it's a small number of reads. And certainly if one was perhaps asking a question independently, under an independence model, if you think this mutation is present, maybe if 2 of the 25 reads are indicated, one might not think it's present, might think that these are just 2 sequencing errors. Okay, but by looking in this structured way, we can sort of see this. The other ones that are there that are interesting are ones where there were just no reads at all that covered that locus. So no reads at all that covered the locus. And Compass indicates that those are not present. We actually impute that they're present. They just were undersampled. Okay. So the summary here is that when you do longitudinal sampling, there's additional signal in the data. And if you're going to be doing this deconvolution problem one would solve in the old days when you had bulk sequencing, or if you're going to do this imputation problem, newer problem when you have single cell sequencing, you can incorporate this longitudinal structure and then get solutions and ask whether or not we really think, clones disappeared or reappeared. And we think we can recover these rare clones by using and modeling this structure. Okay, so in the last several minutes here, I wanted to switch gears and talk about spatial tumor evolution and talk about this at 2 scales. So one is at the scale of cells migrating between different anatomical sites, and one is at the micro scale within one tumor. So sort of understanding when and how cancer cells metastasize is sort of, a really important question in cancer. And we could ask, for example, is a metastasis founded by a single migration, a single event where a cell left the primary tumor, migrated to a distant anatomical site and started that tumor? Or is it perhaps a result of multiple such events? And so this is called whether the metastasis is a monoclonal, there was a single founder clone, or perhaps multiple clones or polyclonal. And so, I think the prevailing assumption until 10 or so years ago was that all metastases were monoclonal. But then once there was sequencing data, cancer researchers started to look into this, and they started to actually try to infer past migrations. Okay? So when they did this, they started reporting all sorts of crazy stuff. Here's a prostate tumor where each of these arrows is a migration, an inferred migration from the primary tumor in the prostate to various other anatomical sites. Another patient, another one, you can see all this crazy stuff. Breast tumor, the sort of what they call multiclonal seeding, polyclonal reseeding, where cells come back to the primary tumor of origin. And so migration happening all over the place through the bloodstream. And so we were interested in looking further at this. And so the problem is actually very simple, one of the simplest phylogeny problems for figuring out the minimum number of migrations. So here's the setup. If I have this clone tree, and let's say for now I've solved all the problems in the first part, so my data is, my tree is perfect. I also know the anatomical location of each cell at the present time, okay, indicated here by colors. So now what I'd like to do is just infer the past migration process. And if we assume that migrations are rare, we can do this under a parsimony setting. And so we just want to compute the minimum number of changes in color if we label the ancestor. So here I've labeled everything with the green color, and that gives me a certain number of migrations. I can count them up. There's the total number of color changes here is what? 1, 2, 3, 4. And so I could, compute the minimum number using just the classic Sankoff-Fitch recurrence, dynamic programming. This was actually even described in the setting of migrations between different locations on a landscape by Slatkin and Madison. And so that's sort of straightforward. But now, from the cancer context, we actually care a lot more about what these migrations were. What was the structure of them? Was it reseeding? Was it polyclonal? We don't just care about the minimum number. So if we want to ask that question, answer that question, we can look more in detail. So if we think about the migration problem, every time I infer an ancestral labeling, so here is one, I can then collapse the migrations that have happened into this directed graph. Okay, so this directed graph just summarizes, there's one node for each location, each color, and there's a directed edge every time there was a migration on the corresponding labeling. Okay, so this is a directed multi-graph because there could be multiple times that the same migration between two sites happen. And so these migration graphs give us sort of a language for describing all this terminology about whether a metastasis was a result of a single event, multiple events, whether cells left and returned to the primary tumor, reseeding. And we wrote down this whole taxonomy in a paper we published in Nature Genetics in 2018, where we talked about, if you look at the graph, if the graph is a tree, then every anatomical site was a result of a single seeding. This is monoclonal, but there's other ways it could be, have migrations with multiple sites. It could have directed edges. This is the so-called reseeding. And so we wrote sort of all of this down. And then we asked the question about, could we infer the number of migrations under a certain one of these scenarios? So you can ask, what's the minimum number of migrations? But what if I asked, what's the minimum number of migrations for there to be reseeding, for there to be a directed cycle in this graph? So this gives us what we call the structured migration problem. All right, so this is, okay, let me try to describe this quickly here. So if you have a labeling of the ancestral nodes, you can collapse that into this directed graph. And so what we're interested in is directed graphs with a particular vertex set, this, original labels or colors. So I could ask about directed graphs on this 3-color set. And I could say, what's the minimum number of migrations if I require that this graph has a certain structure? So what if I require that this graph is a tree? Then what's the minimum number of migrations such that the corresponding migration graph is a tree? Or what if I require that it has a cycle, or it's directed acyclic? Then what's the corresponding minimum number of migrations? And this is something that we worked out in that same paper. So if you were given this clone tree, leaves labeled by these anatomical sites, and you were given a subset of these migration patterns, then you could ask, can you find the corresponding labeling that has the minimum number according to one of these structural constraints on the migration graph? And this was a fun problem. We showed some nice results on some of the cancer data at the time, and it sort of sat there for several years. And remarkably, somehow this year has been the year of people returning to this problem. And there are now multiple papers this year about looking at these migration graphs again. One of them from Muhammad Al-Kabir, who was my former postdoc on the original Machina paper and has something he calls Mach 2. And we created one that we call Fast Mach with a graduate student, Henry Schmidt. I wanted to show you this problem and how we thought about it in this new way. So we went back to the original recurrence for computing the minimum migrations. Okay, so this recurrence really is just very, simple. You sort of percolate labels up the tree, you find the minimum number at the top, and then you percolate those labels back down if you want to find the ancestral labeling. So that works. That will find the minimum number of labels. But now if you impose this global constraint on what the graph will be, that breaks the local structure that these recurrences rely on. So what Henry did was he thought about what happens in computer science when they sort of work on some of these algorithms. There are network flow algorithms that have recurrences. There are all sorts of problems on trees and graphs that you can solve with the structure in the dynamic programming way, but you can also actually write down linear programs for these corresponding problems. And so what he wrote down was a linear program for the Sankoff-Fitch small parsimony problem. So this labeling problem. And interesting, didn't seem that anyone had written down this linear program before. Now, it's not hard to take a recurrence and write it down as a linear program and write down these linear constraints. But the trick is to do it in such a way that you don't have too many constraints. And so what Henry was able to do was write this down in a way that the linear program only had variables that were the number of leaves and the square of the number of colors. Okay, so it's still polynomial time. So then the way he did this was he has this combinatorial structure called the tree labeling polytope. Each vertex in this polytope corresponds to a labeling of the ancestral nodes. Okay, so then you want to find the minimum number. So you'd have this sort of linear constraint that intersects with this tree labeling polytope. So that's nice, but that's just the original Sankoff problem. So what can we do? Well, now once we have a linear program, we can actually impose these other graph constraints as additional constraints in the program. And these will lead to things that give us these various forms of mixed integer linear programs. And so the linear programming version, like Sankoff's algorithm, is efficient. You can solve in polynomial time, but now you can impose all sorts of structural constraints and still be in the realm of linear programming, integer linear programming now. So integer linear programming, these constraints are integral. You may not actually get solutions in polynomial time. So we can go back then and redo everything much faster. So these integer linear programs in this formulation are orders of magnitude faster than what we had for the original Machina, which allows us to scale to much larger trees and inferring migrations. And these much larger trees are coming from single cell sequencing experiments, and interestingly also from lineage tracing experiments here in mice. This was done via lineage tracing of a mouse xenograft. So now the edits are being induced as the tumor grows via CRISPR. This tumor in mouse metastasized from the lung to the liver, et cetera. And so now with hundreds of cells here, we can infer this migration graph. If you ask for what's the minimum number of migrations between these anatomical sites, you get 38 on this tree. You can see the counts here, the number of times you've gotten a migration between each site. But now if you ask, well, what if I want this to be a DAG? So there's no cycles in it. Then the number of migrations, the minimum number, is 41, only a little bit higher. So under some likelihood model, this may not be that much less likely. However, if you ask for the migration graph to be a tree, now the number of migrations goes considerably, and 61. So you can start to impose and ask these structural constraints on the problem. All right, so that's the summary of this part. So looking at spatial evolution across the primary tumor and metastasis. So Machina is the older work. we've now gone back this year and actually made this in a framework that puts this in first a linear program for the Sankoff algorithm, and then an integer linear program when you want to impose any structural constraint on the migration. And this gives you an algorithm that in practical terms is just much faster than we had in the past. Now, I think I'm running up against the hour. So do I have 5 more minutes to do the spatial one, or should we end here?
Sure, go for it.
Okay, so I'll give you the really quick version of this.
Yeah,
There's
awesome.
not as much technical content, because this has been one of the things that I have been wanting to do for years. And the idea here is we know that the tumor grows in physical space. So how we're looking at the micro scale right into the tumor itself. And we've done, we and others, all this work where we think about tumor evolution from DNA sequencing. But wouldn't it be great if we could also see where each of those cells were in space? Now, the good news is there is spatial sequencing now. The bad news is that spatial DNA sequencing is still relatively poor. The technology is very hard to get high quality spatial DNA sequencing. But there is decent spatial RNA sequencing. So what we did was we used spatial RNA sequencing. This is spatial transcriptomics. And without going into a lot of details here, what you can measure is thousands of spatial locations in the tumor, the expression of every single gene. So you're measuring the mRNA for all the genes along with the corresponding location. Many technologies, very rapidly moving area. But the coverage here, there's 2 problems. We're not measuring DNA, we're measuring RNA. So we're only measuring the genes that are expressed. And the coverage per spot is really low. So we can't reliably detect single nucleotide mutations. But what we have a hope of detecting are large copy number changes. So gain and loss of chromosomes, parts of chromosomes. And we can do this because if there's a deletion of a part of a chromosome, then the genes in that deleted region tend to be underexpressed. Or if there's an amplification, the genes in that region tend to be overexpressed. However, it's really, really complicated to actually reliably get these copy number aberrations from all the other variation in gene expression data. So you need to do this in a very subtle way, and there are methods that have done this. And we first developed just a method to do this from spatial transcriptomics data, where we use spatial correlations to help us. And then we also identify the number of copies of the 2 different parental alleles. So we distinguish the number of copies of the maternal and the paternal allele. So now our evolutionary model becomes one after we've run the algorithm, which I won't tell you about, but then what we get is what we call a copy number profile for each spatial location, or actually it's groups of spatial locations because we can't do this at the single spot resolution. And so what that is pairs of integers that tell us the number of parental copies of that segment. Okay, so in light gray here, there's 1 copy of each parental segment. That's the usual case. We usually have 1 copy of our maternal and 1 copy of our paternal chromosome. Every other color here is indicating some gain or loss of one of the 2 parental copies. So now from these allele-specific copy number profiles, we're actually going to infer an evolutionary model using one of the strongest signals in the data, which is we're just going to look at when there's complete loss of one of the 2 parental alleles. So look at these cases where you've either lost the maternal or the paternal chromosome. And we do this for 2 reasons. One, these are the most reliable copy number aberrations to detect. The whole chromosome's gone, we can detect that pretty reliably, one of the 2. And then these loss of heterozygosity events, the loss of a paternal chromosome, is also an irreversible state. Once the tumor's lost the paternal copy of chromosome 17, it's not going to get it back. Okay, so then we can write down a model that just is specific to these loss of heterozygosity regions. And this is just a multi-state irreversible model, sort of a generalization, if you will, of the classic Kamen-Sokal model. So you've got a single ancestral state. In our case, that's the 1-1 state. And then you can lose any of the parental alleles, and these are these derived states. Okay, and there'll again be this thing that you could only lose once, okay, and you never go back. So this irreversibility. We actually developed this for a different data set. I'm not going to tell you about that, but you can go see that in the paper. But now we can use this model to build trees from these copy number profiles. And so here's just one example, and I'll just close here. What we inferred, and I didn't show you this, was 3 allele-specific copy number profiles from the 3 different regions of the tumor. The tree we built, it's a little bit silly almost, because the tree is only on these 3 clones, but we can place these LOH events reliably on the leaves of this tree. And then we can run just a classic phylogeography model in space. Here it's a Gaussian diffusion model. And then we can infer the ancestral locations of the tree. So we can sort of project the tree into space. And so what we see here is the green here is the ancestor here at the beginning here, and then it sort of branches out and grows in these 2 directions. Okay, so the ancestral tree, the tumor growing out into. We can actually do this in 3D as well, because we sometimes have these different sections. And so we're now looking a lot at tumor evolution in space using a variety of different data sets. So I'll stop here. I'll say, cancer evolution, I think sort of fun from the algorithms and computational perspective, because there's all sorts of variations of some of the classic problems due to measurement challenges, due to like this longitudinal sampling, due to the changes, the different markers that you have, copy number aberrations, as I showed you. Phylogeography, I think really interesting for looking at the micro scale as well as metastasis. And for the future, I mean, much higher quality data is coming, especially spatial data, doing whole genome sequencing of single cells coming, looking at non-genetic evolution. You'll actually see this, I think, in the next talk by Trevor Graham next month. And then going from phylogeny to sort of more population genetics models, or things that are all sort of happening now and I think coming more in the future. And I'll end there. Thank the folks who did the work, who are all listed here on the slide, as well as funding agencies who have been very supportive of our work over the past few years. And all the papers you'll see on there and the software to run these things is all there. And feel free to reach out to me if you have questions about any of the work here. So thanks very much.
Wonderful. Thank you. Wow, quite a span of work. Well, we are close to time, but I'll just ask one question. It's interesting, like these ILPs have been, I mean, these are like, you've been working on these for a while, but it was interesting, the juxtaposition that like, there's an ILP approach, and then there's sort of the like probabilistic and like marginalization, and also sort of like, I mean, it's a little bit heuristic, right? Like yes. We like bouncing around tree space, trying moves. And I would say that like, although ILPs have been really interesting, like we haven't seen them really take hold in phylogeny. And so can you, I mean, so now you have this cool new
yes.
where you're using the polytope and you have like these linear programs and it's like simpler. Is that something that is sort of, you think is going to be of more general interest, or is there, I don't know. I mean, It's not really powerful.
Yeah, no, it's a great question. I think one of the things we've been excited about. So with the polytope, we can do like the maximum likelihood version, because that's just like weighted parsimony. But when you start doing marginalization, it gets tricky, and that's where that polytope thing falls down. But we've been really interested in, we're starting to kind of think about, when we do the ILPs, we're really modeling like the structural constraints, right? And so that creates these sort of structural classes of solutions. And we did this a little bit with the longitudinal work, where we sort of think about if you can impose these structural constraints and then do some sampling under these constraints, then it might be a way to do something like importance sampling. It might be a way to do, what we'd want to do is in the metastasis setting would be something like, oh, well, could you tell me like the probability that there would be a directed cycle in this data? Mm-hmm. And that's like a structural constraint. It's hard to get at that because you have to like sort of marginalize over all solutions where there were cycles and and then the rest of them. And so we've been thinking about that. We think there's some, games, there's something to do there, but it's tricky. I mean, I think, when you get into MCMC, it's like thinking it in terms of like important sampling or constraining the sampling space according somehow to these combinatorial constraints.
Yeah, I would love that. That would be so cool if we could have some sort of limited marginalization that is like a lot smarter than just sort of let's try everything.
Yeah. So, it's difficult, but I think there's some opportunities there.
It's so cool. I mean, polytopes are amazing. I was like, when I saw that slide, I was like, oh, ooh. I'll have to check out that paper. Well, thank you for that. Much appreciated. And like you say, next time we're going to have Trevor Graham. So
yes.
Thanks very much. And yeah, thanks for participating. It's really neat.
You're quite welcome. Thanks for the invitation. It was great to speak to the group.
All right. See ya.
Bye-bye.